grad student from University of Manitoba, Canada. Today I'm here in the ASPB 2030 to present my poster. It is a role of some predicted rat-core interacting proteins and then their role in UV damage DNA repair. So basically, in case of animal model, the UV damage DNA repair is a known process, and then for the DNA damage recognition step, there are two complexes that are involved in it. One is DDB1, DDB2 complex, another one is XPC HR23B complex. So it is well understood in human system, but not in plants. XPC has a homologue in plant system, which is called RAD4. So we were interested in RAD4, and then we started looking for other interacting proteins with RAD4 as well. So in the Arabidopsis interaction viewer, we found that two positive control, these two like RAD23B and then XPB2, which are already known to be interacting, like predicted to be interacting with RAD4. And along with that, surprisingly, we found two uncharacterized protein with it. So we were interested in these two proteins, which is basically RNA-like superfamily proteins. So we just call them RNI1 and RNI2. So we ordered tDNA insertion lines, lots of function lines from ABRC, and then we started working on those. And then we did UV sensitivity assays on those because RAD4 homologue XPC is found to be basically recognizing the DNA damage in animal system, so it might have some relation with UV damage DNA repair. So we did adult assays, and then we sibling assays with those loss of function lines of these interacting proteins, and both of them are found to be sensitive to UV. So we also raised overexpression lines, and then under the microscope we found that these YFP proteins, we see some punctured nuclear patterns under the yellow filter, but in case of red channel we didn't see anything. So these act as a background. And then finally, because we started working with those interacting proteins, so we wanted to see those proteins actually interacting. So we raised these, we performed these two hybrid experiments with this RNI2 and RAD4, but unfortunately we didn't see any interaction though. We have seen those spots in the control, but in treatment in this quadruple dropout media, we didn't see any spots. So those two are found to be not interacting with each other. But also, we just we started working with RNI2. We are hopeful there might be something going on with RNI1 too.
Wonderful, thank you.
Thanks.
How are you enjoying the meeting?
Oh, I'm super excited. This is like my first time, and then I always dreamt of joining ASPB sometime, and then finally I met it. I'm super excited. I'm super happy. I like it so much.
Wonderful. And do you think the society could do something to improve or serve you over the next meetings?
It's hard to say because my first meeting, so I was so excited. But what should I say? This is pretty organized, I found, because I have attended other meetings, and I know ASPB is really big. Other meetings might be what I did join is not that big, so pretty organized. And then with a lot of people, it's a little hard to organize such a big conference, right? So I think you guys can be more organized. That's all I can say. Otherwise, I really like it.
Wonderful. Thank you.
Thanks.